Hello everyone, this is Sean Taylor, Field Application Scientist Manager for GenScript in North America. And in this seminar, I'll be discussing a novel assay technology for the detection of neutralization antibodies to COVID-19 and its comparable performance to other currently available serological assays for coronavirus. Now, when we discuss the test kits that are out there, we need to understand the course of infection of COVID-19 or any virus. And when we look at this chart down here, we can see that when a person is infected with a virus, the first detectable molecule that sheds into the person's nasal passages or mucus is the viral RNA from coronavirus. So we're able to detect this with an assay called qPCR. So this would be for very early detection. But then as the immune system takes over, our own immune antibodies start to be released to fight the virus. And it starts with the release of IgMs and then IgGs for longer term immunity. So there is a temporal aspect to detecting the virus that relies on the different molecules that are released and our own immune response to the virus, which comes later. So qPCR is typically what's used first for early detection, followed by serological assays to detect IgM and IgGs later after an immune response. Now, there is a complementary aspect to these two tests. So for nucleic acid testing, as I explained in the previous slide, it's very sensitive to the presence of virus very early after infection because those are the molecules that are released by the virus initially. So when we're initially loading up with virus from infection, it's the viral RNA that can be detected, and qPCR is a highly sensitive assay for doing this. A couple of drawbacks of qPCR is that it is dependent on sampling. It's critical that the sample be taken from the correct portion of fluid, either deep in the nasal passages or a back in the throat swab to catch virus shedding from the deep lungs, because that's where the virus likes to live. And when we cough that virus into the back of the mucous membranes in the back of the bronchial or nasal passages. So sometimes we can get a false negative simply from not taking the sample correctly. Another problem with qPCR is that because we're looking at RNA from the virus, RNA is very easily degraded. And it's important, therefore, that the sample be stored in a solution that preserves the RNA for a long period of time between sampling and testing. And sometimes if those solutions are not well looked after or well controlled, then the RNA that we've collected can degrade before being tested, and that will lead also to a false negative if the RNA is degraded. But if we're able to manage sampling and storage of the sample, qPCR is an excellent technology for reducing disease period, which is the time by which an individual is unaware that they're infected and able to infect other people, and therefore they can find out quickly if they're infected and self-quarantine. Serological assays, as we discussed before, are a consequence of a natural immune response. So in order to be able to test the immune response antibodies, IgGs, IgMs, IgAs, an individual has to have recovered from infection or at least is in the phase of recovery. So this takes more time. Typically those antibodies are only detectable at least a week or so after the onset of infection. But because of the timing, so if we go back to this slide, we can see that if we take a sample early in infection where there's a lot of viral RNA, we'll get a positive qPCR test, but we'll get a negative for the serology test because we haven't initiated the immune response yet. And vice versa applies. If we take a sample that's later after infection, all the viral RNA is gone because we've cleared the virus. The virus is no longer circulating in our system, and all we have left are the immune response antibodies that persist after infection. So you can have a positive serological test and a negative PCR test or a positive PCR test and a negative serological test. So timing is everything for getting a positive result with these types of tests, and therefore these types of tests are very complementary. It's typically recommended to have both tests, and if one test is negative. If we have a negative PCR test, it's important to still take a serological test just in case you were already infected, perhaps asymptomatic, you didn't know you were infected, and the serological test will uncover if you have the immune response antibodies indicating you were infected, and then contact tracing can happen to go back and see the individuals that you interacted with to know if they were infected as well. So the currently available tests for, most popular tests at least, for detecting COVID-19 serologically are the ones that detect IgGs and IgMs specifically. So in these tests, what we're looking at is using proteins that our immune response will respond to rapidly. And those proteins include the spike protein. So this is a cartoon image of the coronavirus with the Spike-glycoprotein protruding out of the virus. And the Spike-glycoprotein has a receptor binding domain right at the very tip of the Spike-glycoprotein. This is highly antigenic, and a lot of immune response antibodies are generated to this. Within the virus is the nucleocapsid protein, which encapsulates the RNA, and that's also highly antigenic. So we generate antibodies to both of these proteins, the receptor binding domain as well as the nucleocapsid protein. Now, when the virus interacts with the host, it binds to what's called the ACE2 receptor. So this represents the lipid bilayer of a host cell, where we have the ACE2 receptor coming out of the lipid bilayer membrane outside of the cell. And this is what the virus binds to. And when the spike protein and the receptor binding domain interacts with the receptor, the ACE2 receptor, then the virus is encapsulated, endocytoses inside the host cell, and then it replicates itself. And that's how infection progresses inside an individual, by this process of the virus binding to a host cell via the spike protein of the virus and the ACE2 receptor, and then going inside the cell, taking over the host cell machinery and replicating more and more virus. So during this process, as more and more virus circulates in the body, the immune system begins to take over, and we end up getting immune response antibodies. So immune response antibodies to nucleocapsid protein, and immune response antibodies, again, IgGs and IgMs in both cases, to the receptor binding domain. There are immune response antibodies that can happen to other regions of the spike protein. We're going to focus on the receptor binding domain because this is the most popular protein that's used in detection assays. So what people have done then is they've sequenced the RNA from the virus to determine what is the exact sequence of the receptor binding domain, and then we've been able to produce this in bacteria. So we have receptor binding domain protein that we can purify, and same thing with nucleocapsid protein. So these are now purified proteins that we can coat on the bottom of ELISA plates. So this is the most popular ELISA that's out there for detection of IgGs and IgMs circulating. It's by coating the bottom of an ELISA plate with the receptor binding domain. And by doing this, when we incubate a serum sample from an individual, the immune response antibodies, as you see here, will bind to this receptor binding domain. So they'll bind to the receptor binding domain, either IgGs or IgMs, and then you add a secondary antibody, a mouse anti-human IgG or IgM, so this could be either one, which will bind to the immune response IgG or IgM, and then we can get detection. So this is a very commonly used format for ELISAs that people are using currently commercially and homemade kits to detect the immune response antibodies from COVID-19. In this version, we have the receptor binding domain and the nucleocapsid protein bound to the bottom of the plate, so we can detect more of the immune response antibodies, because again, there are not just immune response antibodies to receptor binding domain, also to nucleocapsid protein, so we have more immune response antibodies bound, which might augment the sensitivity of the assays. But again, it's the same principle. We're using the purified proteins from the virus itself, either receptor binding domain or nucleocapsid protein, bound to the bottom of a plate. We incubate the sample, the immune response antibodies will bind to these proteins, and then we can detect with the secondary antibodies to those immune response antibodies. So these are very commonly used kits out there, currently used for detecting IgGs and IgMs. There is a new novel assay that GenScript has released. This is very new, interesting type of angle at detecting the immune response antibodies. So again, just to show, we have the virus binding to the ACE2 receptor, which would be internalized. If the antibodies bind to the receptor binding domain, the spike protein here, what they'll do is they will block the ability of the virus to bind to the ACE2 receptor, and that's called neutralization. So these antibodies, in effect, are neutralizing the ability of the virus to bind to the ACE2 receptor. And therefore, if the virus can't bind to the ACE2 receptor, it can't be internalized into the cell and it can't replicate. Therefore, you've effectively blocked the ability of the virus to propagate, and it will just die off. So these immune response antibodies that bind to the spike protein, as you see here, are really important, and they're called neutralization antibodies. So what we've done is we've created an assay to detect these antibodies. The currently available assays that are out there are a traditional virus neutralization test, a VNT test. And in this format, we use a live coronavirus. So this would be in this version of the kit, you would have a live COVID-19 SARS-CoV-2 virus, which is highly infectious. So we need to use a BCL level 3 lab to do this, and appropriate personal protective equipment to be able to protect us from infection to that virus. So this is a very dangerous type of test. And you incubate that virus with the patient's serum sample, which contains the antibodies. And in the case where the patient doesn't generate neutralization antibodies, the virus will bind. And we also have cells that express the ACE2 receptor. So these would be cells that express the ACE2 receptor. So you mix the virus with the patient's serum to assess the antibodies, and then you mix that with the cells, and if you get binding, that means there's no neutralization antibodies. However, if the patient is generating neutralization antibodies, the virus will not bind. And the readout here is a luminescent reaction. So if the virus binds, you get a luminescent light emission to detect, and if they don't bind, then there's no light emission. So the advantage of this test, of course, is that it perfectly mimics the infection response inside an individual. But the disadvantage of this test is that it requires a very specialized lab, very expensive equipment, and personal protective equipment as well. And it also requires a lot of time. This is a 2 to 4 day test, and it takes highly skilled, highly trained people to perform this test. Now, there is another type of test that's a little less, requires a little less safety equipment than the initial virus test, and it's called a pseudovirus neutralization test, a PVNT test. In that test, what we use is what we have here, a lentivirus. So this is a lentivirus, which is an innocuous virus that expresses the spike protein from COVID-19. So we're still expressing the spike protein, but we're using a virus that's not infectious. So we can use a BCL2 level lab, a much lower level containment facility with less protective equipment, because we're not using COVID-19 in this case. It's the same principle though. We incubate the pseudovirus, the lentivirus, with the patient sample, and if we get binding, we get luminescence, and if we don't get binding, it's because there are neutralization antibodies in the patient sample that are blocking the binding to a cell line that expresses the ACE2 receptor, and we will not get luminescence. The advantage of this assay, of the pseudovirus assay, is of course that it's safer. So we don't need a BCL3 lab, we don't need a lot of protective equipment and training and highly skilled technicians. We need a much the lab requirements are much lower in terms of safety. However, it still takes a long time. It's a very long test. It's laborious, 2 to 4 days. GenScript does provide this as a service, though. And GenScript also provides this material, lentivirus expressing spike protein and HEC293 cells that express the ACE2 receptor, as a kitted assay. So if there is a desire to perform this kind of a test, GenScript can offer it as a kit or as a service. So it kind of eliminates the upfront work or downstream work required to perform this test. The third test, and this is the novel test, this is new from GenScript, is what we call the Surrogate Virus Neutralization, or cPass test. So we abbreviate this SVNT. This is a brand new GenScript product, and this is a basic ELISA kit. So what we've done in this kit, and I'll explain this in the next slide, is we've taken purified RBD, purified receptor binding domain from the spike protein. So again, the same protein that's used in traditional ELISA kits actually. We've conjugated that protein to horseradish peroxidase. So we have RBD conjugated to horseradish peroxidase, and we've purified the ACE2 receptor, the human ACE2 receptor in cells that the spike protein would bind to, that the receptor binding domain effectively would bind to from the Spike-glycoprotein. We've coated plates with the ACE2 receptors. You can see this green ACE2 receptor is coated at the bottom of the plates, and that permits the use of just this receptor binding domain protein incubated with patient samples to assess if there are neutralization antibodies expressed in the patient samples. So let me explain this in more detail. This assay was invented by Dr. Linfa Wang, and the article that describes this assay is published down here. So you can read this. It's still a preprint version, still in review, but it's available online. So you can click on this link down here, and you can review the article in more detail. So how does this assay work? So what you do is you mix the receptor binding domain conjugated to HRP. So I've abbreviated this RBD HRP. So receptor binding domain, and the dot in blue is horseradish peroxidase. So you incubate this purified protein, which comes in the kit in solution, with the patient sample. So with samples that contain antibodies. This would be great for vaccine development, where we're trying to look at antibodies expressed to a vaccine, and we can again incubate those antibodies with this RBD HRP complex. So we take the sample, we take our RBD HRP, and we incubate those two together. So this is what this image would be here. This upper image is just the incubation of a patient sample which contains antibodies to RBD, and the RBD itself, RBD HRP. This is a 30-minute incubation at 37 degrees. And then after this incubation, we add it to the plate that's coated with the ACE2 receptor. So I've denoted neutralization antibodies, so antibodies that block the binding of the RBD to the ACE2 receptor in red. And in blue, I have other antibodies that are just non-neutralizing. They bind to regions of receptor binding domain that don't affect its interaction with the ACE2 receptor. So of course, in this case, we would have 3 molecules of the RBD-HRP that are effectively blocked by the red neutralization antibodies, and 1 molecule that isn't blocked. So it can bind, so that's what this arrow denotes. So this 1 molecule can bind to an ACE2 receptor. So we incubate, this is about a 15-minute incubation, and then we wash the plate. So when we wash, we're going to wash away, obviously, these complexes that are the red complexes because they're blocking the ability of RBD to bind. So they'll go away, and the 1 complex where there wasn't a red neutralization antibody will bind. So this figure here is just to remind you that this molecule here, this brown molecule, is the receptor binding domain, and this receptor here is the human ACE2 receptor. So we're mimicking the binding of the virus, if you will, to the ACE2 receptor by just using the purified proteins. So after we've washed the plate, we then add TMB, and we'll get a light yellow color because there's only 1 molecule bound here. There were a lot of neutralization antibodies in this case. In the lower schematic we have here, we've added, again, a patient sample, but this sample didn't contain neutralization antibodies. So now we mix the RBD-HRP, purified RBD-HRP, with our sample, and there's only non-neutralizing antibodies in the sample. So all of the RBD-HRP complexes, therefore, can bind to their associated ACE2 receptors, as we see here. So therefore, we have a lot of binding, and obviously it's HRP that makes the TMB yellow. So all these HRPs now that are here in blue will make the intensity of the color much darker, as we see here, in comparison to up here where we only had 1 molecule bound. So the less neutralization antibodies we have, the darker the yellow color. In here we had no neutralization antibodies, no red antibodies in the bottom schematic, so therefore a dark yellow color. And up here we had a lot of neutralization antibodies, so we get a lighter color at the end of this reaction. So a very simple test, about a 30-minute incubation initially with the neutralization antibodies, then about another 15 minutes or so with the complex mixture, and then a wash, and then we add the TMB and we read. So it's a little over an hour, this test to perform. So what are the advantages of this cPass SVNT assay for detecting immunoglobulins? So the first advantage, which is really important in comparison to other commercial tests that are available, serological tests, traditional ones where we have, as I mentioned, the RBD bound to the bottom of the plate, is that it's immunoglobulin agnostic. So because we have RBD in solution in this case, in this test, any immunoglobulin that we generate, any IgG that we generate to COVID-19 will bind to the receptor binding domain and will likely block its ability to bind to the ACE2 receptor. So we're able to have a much more sensitive test because in the opposing tests where we have RBD bound, we need to detect with a secondary antibody that's very specific to the IgG or IgM or IgA, whatever class of immunoglobulin that we're trying to detect. Here, we're detecting whatever immunoglobulin to COVID-19 binds to the receptor binding domain. So it's a more, in theory, it's a more sensitive test. The other advantage of this test is that it's species independent. So because we're using purified receptor binding domain from COVID-19, from SARS-CoV-2, and we're using human ACE2 receptor, if we're working with animal models where we've infected the animals with COVID-19, then the antibodies that they generate will be to the receptor binding domain, and they'll block the ability of RBD to bind to ACE2. So we can use this test for any type of animal model as well, from small animals all the way through. So if we're doing preclinical or clinical trials for vaccine development or for a drug, an antibody-based drug, to alleviate the symptoms of COVID-19, this is a great test because it permits the ability to use a very consistent test from the beginning of using mouse models or even cell-based assays all the way through primates into humans. The other advantage of this test is that the RBD is in solution. It's not coated on the plate. So as we see, this kit is provided with purified RBD. So the advantage of that is that the RBD is in a much more structurally like conformation to what it would be in the human body or in the infected state circulating, as opposed to the assays that have coated RBD on the bottom of a plate, where the RBD, by being coated, could have changed its conformation. The effect of coating RBD onto the bottom of a plate could change its conformation and expose antigens that might bind to other immunoglobulins unrelated to SARS-CoV-2, or might perturb the ability of the SARS-CoV-2 antibodies from binding, because those antigenic sites may have been altered by coating at the bottom of the plate. And this is where we hear a lot of the assays that use RBD coated on the bottom of a plate there are complaints about specificity and sensitivity. And although this is not proven in the lab at this stage, there's a lot of speculation that the coating of RBD itself could lead to these issues of specificity and sensitivity. So in this assay, we have native RBD in solution, so much more structural integrity of the RBD in solution to mimic the correct conformation for antibody binding, which augments the specificity of the assay. So here, because it's immunoglobulin agnostic, we've increased sensitivity because it binds all the immunoglobulins, and down here, because there's a lower probability of conformational differences in RBD, we have potentially much higher specificity in this assay for the immunoglobulins to interact. Now, there's also the advantage of this assay by being a dual detection and functional assay in a really fast test. So as I mentioned, this test takes a little over an hour to do for a 96 well plate. So it's a very fast test. And it's functional because we're not only detecting the immunoglobulins bound, but their ability to block the binding to the ACE2 receptor. So we're getting a lot more confidence in our test that these immunoglobulins are functional immunoglobulins that neutralize the ability of RBD to bind to the ACE2 receptor. So we're getting more information from this test as well. So now, just to show some data here, what we did was we took difficult samples. So this is not a full study cohort of samples. We took very challenging samples from a number of different study cohorts that were performed with IgG and IgM tests. And we took all those challenging samples, or several of them, which came up as being the wrong call with IgG, IgM tests. So we had a bunch of PCR negative tests that came out positive. They should have been negative. And some PCR positive tests that came out negative and should have been positive. So none of these samples were corroborating in an IgG, IgM test. And this was a test where, again, the receptor binding domain is coated to the plate surface. We then tested these exact same samples with the cPass neutralization test. And what we found was that all of the samples gave us corroborating data. The negatives were negative, the positives were positive. And this was quite impressive. We weren't expecting this. So this was a really surprising result, which supports the very high specificity and sensitivity of this assay, likely associated with more immunoglobulins being able to bind to RBD, more immunoglobulins being tested in this assay because the RBD is in solution. And also because the RBD is in solution, it's more conformationally less perturbed. It's conformationally more intact because it's in solution than a coated RBD on the plate surface. Then what we did was we assessed the correlation between the SVNT test and a plaque-reducing neutralization test. So this was only 20 samples, but as we see, the corroboration was 100%. So for these 20 samples, the cPass test, our SVNT ELISA test, was directly, accurately corroborating a much more laborious and challenging plaque-reducing neutralization test, which would require a level 3 lab, especially when working with live virus. So very nice data and supporting the use of this test for looking at neutralization antibodies, and nice data here for supporting the test for just looking at people who were or were not infected with COVID-19. Now, we do have other data from beta testers of this kit, from the beta testing period where we were looking at this kit, that showed excellent corroboration with viral neutralization assays. So there is additional data that we can't show because of the collaborations that we have with those labs, but the corroboration between neutralization tests and this cPass test are excellent. So here, this is just to exemplify. This was data that we were collecting for submission, for EUA submission. And what we did was we ranked our samples by the amount of time post-symptom onset. So if we look at samples, and again, this goes back to this graphic that I showed in the earlier slides, where timing is everything for tests to work properly. The timing of sampling is important. So if we go below 7 days for the SVNT test, or I would say any serological test, then the corroboration between PCR and serological testing will be quite low. So only 74.1% sensitivity, and that's likely because there just wasn't time to generate antibodies yet. So this is not because the test is bad. This is simply because the timing of sampling doesn't give enough time for an immune response to generate the antibodies to detect. So only 20 of the 27 samples that were PCR positive were positive with the SVNT test, simply because of timing. If we look at the samples that were collected after 8 days of symptomatic, where we had 99 PCR positive samples, then we had excellent corroboration with the SVNT test, so 99%. So all these samples gave, except for one, gave really good data. And then we also tested some unknown samples that were PCR positive. Obviously, these samples must have been more than 8 days because they were 100% corroborative. So it's very important when looking at data to assure that the timing of the sampling supports the test to use. If it's less than 7 days, you should probably go for a qPCR test, but if it's greater than 8 days, qPCR will likely give a negative result, and then we go for serological testing. So here was overall clinical data that we collected over a variety of different clinical tests, and we just accumulated all those results. So we had 193 PCR positive samples and 480 negative healthy patient samples. And we tested all of these samples. Now, unfortunately, in this 193 samples, we included the 7 samples that were less than 7 days. So we still included them in this study to be fair with trying to corroborate data with PCR positive data. So the reason why we only have 93.8% sensitivity here has as much to do with the fact that 7 of the samples that were negative were less than 7 days. So this number is actually a little bit artifactually low. It should be much higher than this, probably around 96 or 97%, if we would have added back those 7 samples that were less than 7 days post-symptomatic, because they were going to be negative anyway. For the healthy samples, excellent correlation, 99.4%. So very, very good corroborative data. So if we look at accuracy of this test, the accuracy is the overall probability that a patient will be correctly classified. We have a 99.1% accuracy. So this test is extremely accurate for calling patients as having had COVID-19 or not. So it's a great alternative test to traditional IgG, IgM tests that are out there. The positive predictive value, a little bit lower than we would like, 88.8. But again, this number is lower also because we used we incorporated the samples that were less than 7 days post-symptomatic. So this number should be in the 90s. And then the negative predictive value is very, very high as well. So very solid, excellent test actually. And these numbers are better than what we're seeing out there for traditional IgG, IgM test using receptor binding domain coded to the plate surface. So a really sensitive, accurate, and specific test for COVID-19. We also looked at cross-reactivity. I'm not going to go through all this data in very great detail, but just to say that we tested all the usual suspects for viruses that are out there and coronaviruses that are out there. And the only virus that gave us cross-reactivity was SARS-CoV-1 here in yellow. So SARS-CoV-1, there is cross-reactivity with this SVNT kit. We don't consider this to be a major issue because SARS-CoV-1 is essentially not circulating highly globally. So the test is very, very specific actually, and with very little cross-reactivity. And finally, just a little bit of data on cross-species. This is the data that's published in the article by Linfa Wang. So you can see these results there. It's just to show you that it does work well in rabbits, and the test does show neutralization in ferrets. In this case, in ferrets, they used SARS-CoV because they knew it was cross-reactive. Here they use SARS-CoV-2. We have seen data from other animals out there, large animals and also small animal models. So the test, as expected, given the nature of the way the test is constructed, is definitely species independent. So GenScript has a variety of proteins available to generate tests. So if you're looking for proteins to do research with COVID-19 or to generate kits for detection yourself for COVID-19, then definitely visit the GenScript website and you can go through. We have all the proteins that are from the receptors, from the host cell, and for the virus itself to be able to generate these kinds of tests. We also have the antibodies that would be required to generate these tests, and paired antibodies for ELISA tests, as well as the ELISA kits that I mentioned. So we have also services to be able to build tests for you if you don't have the capacity to make tests. So we have qPCR detection services. Again, the ELISA detection kits that I explained, particularly the SVNT kit, which is now becoming very, very popular globally, the proteins and plasmids to perform research. So if you are searching for solutions for coronavirus and COVID-19 detection, GenScript is definitely a good place to go. This surrogate virus cPass test has bubbled up to the surface as becoming a very popular test. We strongly recommend this as an alternate means for detecting IgGs, IgMs, and neutralization antibodies. And we feel that this will be a game changer for not just detection of past infection, but also for vaccine research and antibody-based drug research for COVID-19. Any questions?